Alright, well, welcome to my presentation on Chapter 8 of not US, AP Bio. In Chapter 8, we are discussing the creation of the double helix models and the people who helped in its creation, and in the gene recombination and expression. So let's get into it. So these two gentlemen on the screen are James Watson and Francis Crick. So they met in 1951. So, for some context, DNA was discovered in the 1860s, but at the time, no one actually was thought it did anything. They just thought, oh, cool, squiggly lines, and everyone thought that protein was actually where genetic info was stored until about the 50s. So, but they didn't know that DNA was made of nucleotides, which in turn were made of sugar and a phosphate and one of the bases listed. So the two decided that they were going to try and create a physical representation of a DNA, and they based their work model off Linus Pauling's, which he made a protein that was a single-stranded twisting helix. So in the next line, I want you to think about these questions: How did Franklin find and present her info, and how did she provide for the creation of the double helix model? So, ro- so Photo 51 is the photo you see on the screen of the circle thing, so it's one of her more famous slides. So in 1951, Watson went to her lecture at King's College in London, and so she was presenting slides, which she had captured using X-ray diffraction. So slides were DNA she had exposed to increasing humidity, and with causing him to elongate. So what she basically proved was giving a basis that, oh, it's a helix, because as it stretched, went out straighter, but unelongated, it's a helix, and that also proved that DNA was inside the helix because of the hydrophilic phosphates found in DNA, and so and she would continue to help later. In 1953, when her lab assistant by the name of Maurice Wilkins had showed the two, since they were working in the same lab, that uh, one of her unpublished slides, that without her permission, of course, and and basically it showed, and Watson said it was greatly helpful because it showed some of the parameters of the helix model that it was two symmetrical chains together, and that the nucleotides had to be opposing each other. Next one, I want you to think about these questions on the screen. Erwin Straff, this is the gentleman inside. So in 1952, he came up with this rule that no matter the species, no matter what, that all the bases in a phosphate will be equal to each other. A will always have the same number of phosphates as T, and C will have the same number of bases as G, no matter what. And also, he provided them an ideal of basically how it basically looked, like texture, like it was uniformly thick and it was a smooth shape. It wasn't like randomly bumpy and stuff. Next. So the two pair finisher model in 53, it was almost immediately accepted by the scientific community because it fit all the experimental data up to then perfectly. And it also helped reveal what how the DNA replication basically worked, which is it unzips, and then one of the helix acts as the strand acts as a template to make a complementary one to be then go to another one and reproduce the DNA. And in 62, Watson, Kirk, and Wilkins all won a Nobel Prize. Rosalind, however, had died 4 years earlier due to cancer, and as such she was not eligible for said prize, which caused a stir of controversy due to some thinking it was just because she was female that she was denied, besides the posthumous part, and that many felt she got the short end of the stick. So, protein folding. So, proteins are all made up of 20 different amino acids. That an actual protein is a linear chain of all of them together, and and then when they're all folded into 3D shapes. And now it's important to note that each shape is called a conformation, and it acts as like the identifier for what that protein does. So as you see in the picture, we got like lines all on together, and then they all crumple up into a sort of sphere almost. What you think about these ones next? About how transgenic organisms are created, and what's the difference between regulatory encoding. So a transgenic is basically it's basically if you put turtle genes in a snake. It's anything with genes that you know aren't usually in said creature. And they use the two sequences, the regulatory, which basically acts as it's the counter in a way, basically how much of a protein is produced by that gene. And then also the coding sequence, which basically specifies what the amino acids are in said protein. Ahem. So then the gene when a gene so after they engineer a new gene, it becomes a recombinant gene, which is basically a gene that wouldn't be naturally found in said creature. And then it's carried by a vector to then lead to gene expression. So for a protein, for no, sorry, for a gene to be able to create a protein, it has to do two steps. Transcription, which as the name implies, basically it makes an mRNA, which then makes it takes a strand, it latches on, and then it sort of makes a complementary strand to that strand to then move over, which it's basically identical to the strand except all T's are turned into a U. Then it travels over, latches onto a ribosome to then be translated and start producing amino acids. So when amino acids are put into groups of three, which are called codons, which are then transferred via a tRNA to latch onto the mRNA with a connector called an anticodon. So codons do one of three things. They either they all specify what the amino acid is, but there are two specific types that are of note. A start codon, which acts as a marker of where a sequence begins, and a stop codon, which basically, as it names, stops a sequence. And basically, codons follow the genetic code to how they are set up, and they have two properties. They are redundant, they're not ambiguous, and they're not universal. And that is my presentation. I want to thank you all for listening, and I hope it's good. Um, and yeah, I hope it was very helpful, and I'll see you later.